Anatomy of gene. So far we have discussed about the methylation and the acetylation, which cause activation and repression of gene expression. So the first thing that basic is your exon and intronic sequences. The exonic sequences are the coding sequences, whereas your intronic sequences are non-coding sequences. So here is this picture. You can see that the region that code part of the probably. Okay, so here's the picture. And the first thing that you see in this picture is the promoter region, where the RNA polymerase II binds, which transcribe this gene sequence. So this is the transcription. The TBP is the transcription binding protein. The gene called beta globin, your exon is leader sequence transcription. So this is the sequence from where the transcription begins to form the nuclear RNA. This is the translation initiation codon ATG, which will be in AUG in your mRNA. Intronic sequences. Then your translation termination codon like UAA, USA, UGA. Exonic sequences, polyadenylation tail, which is in the exonic sequence region of 3' UTR region. And this is your transcription termination region. So this is the basic things. The leader sequence for just some of the site, which is like your anchoring region, which binds. The CG region is in the promoter. Data box is found in the leader sequence, processing AUG, translation keeping. The data box in the promoter region, where RNA polymerase II bound comes and bind. This is the region. Then your transcription incision site cap sequence. This is the 7-methylated guanine capping region, and this is found in the exonic sequence of 5' UTR untranslated region. This is also in exon sequence, like in polyadenylation tail site in the 3' UTR region. And then this is the leader sequence in the 5' UTR region. Then nothing so important. This is the polyadenylation in the 3' UTR region. About 1 to 300 adenylate provides stability, allow this mRNA to exit the nucleus, permits the mRNA to translate into protein molecules. So the cis regulatory elements, the turn on and off of dimer switches of a gene. Here you can see that the promoter region has possessed some CpG island. This is your nothing but the cytosine and guanine intermediate, bonded with the phosphate bond. So phosphate ester bond, which is formed combined this cytosine and guanine in a polymer, in multiple number sequential order. This is called CG isolate, about 1000 base pair reaching the sequence, which print, and these are present in every cell and specifically bind to the CpG rich region where the basal transcription factor is bound. So this is the reason in this transcription binding protein has some promoter, this promoter region where the transcription binding protein, this factor, which I've just told you right now, this is your basal transcription factor, which allows this RNA polymerase to bind with this factor, and this factor has a complementary to that of your promoter region. Then your enhancer and silencer. Enhancer are something different from that of your promoter. As you can see in this picture, the promoter is upstream to that of your transcription initiation region, but still the enhancer is further upstream to that of your promoter. So let me show in this picture. Here you can see this is the transcription initiation site, and its offstream is the promoter region, but the enhancer is far more upstream to that of your promoter region. And some intermediate binding protein, as you can see very clearly, the picture here shown is a promoter region bound with RNA polymerase with the help of some transcription binding protein or some binding protein which will identify that particular sequence in the gene, and that allow this RNA polymerase to bind with that region. And some intermediate like LPD1 transition factor, these are all transition factors. Some associated, what we also call associated factors. These intermediate factors bridge the bond between enhancer and promoter, and this allows this faster and this protein requires the nucleosome modifying enzyme to transcription associated factor and stabilize this RNA polymerase 2. Here it's written. It stabilizes this RNA polymerase 2, and this allows this transcription initiation of that genomic sequence. Now, nothing so important. Well, here acetyl group attached, that's inhibit the transcription. That's it, nothing. Enhancer modularity. The genetic, you can see this is a picture of experiment which is done, which shows the genetic elements regulating the tissue specific transcription, and that can be identified by using your reporter gene such as your enhancer region of regulating process protein can be tagged to show this expression of the genetic tissue specific transcription being bind. So here's an example, very nice example of enhancer. So multiple enhancer in a gene can be there. As we know, we don't have a polycystin type of gene, but we do have some combining effect of exonic and intonic, exonic sequence to by means of spliceosome to remove those introns, and we have a combination of those exons to multiple types of protein can be extra. I will show you. Okay, so this is here in this case. The basic brand specific enhancer, the brand specific enhancer and the lane specific enhancer is there. So whenever there is a specific transcription factor related to brands goes and bind with that factor, it with some help of some intermediate associated transcription factor, it stabilizes this RNA polymerase 2 and cause a transcription of a particular type of mRNA being expressed on this brand region. And when the limb express transcription factor goes and bound, it will bound to that region of enhancer where you can see, for the example here, the transcription factor has bound with this blue color enhancer region, and where this limb specific enhancer is left without any bindings. Similarly here in this case, this region brand specific enhancer not being used here, whereas your limb express transcription factor is used, and this causes the formation of your mRNA expressed inside the limb region. And this is the thing, this is called the enhancer region modularity. This is specific. So this is how the gene are being regulation by means of enhancer. Then this is combinatorial association, although there is a modularity among enhancers. So let me show you the picture. This is a promoter, this is an enhancer region. So whenever there's some packs, this kind of thing, when the promoter and enhancer, you can see the lens, it's some gene activation. So whenever the enhancer is being bound with some promoter region, then only it will be cause this activation of this gene to be get activated, or else it won't. And here's some lectures experiment, not so required. This is some family of transcription factor families with subfamilies. You can check this out. Okay, so the transcription factor domains, there is a DNA binding domain that recognizes a particular DNA sequence in the enhancer. There are several different types of DNA binding domain. Transactivating domain, protein interacting domain. So this is 3 domain that found in transcription factor. I will show you a picture. Here it is. This is a picture of a 3-dimensional model of a homodimeric transcription factor, and as you can see here, There are three regions, as I've told you in this opposite. And the three region is your DNA binding domain. This is in this picture, you can see the DNA binding domain is here. This recognizes a particular DNA sequence. The second one is your trans-activating domain that activates and suppresses the transcription of the gene whose promoter enhancer is bound with. So in this picture, it's not. This is your amino terminal, but not shown here. But here by it helps to bind with the what is written here. Thus, that activates the suppress the transcription of a gene whose promoter enhancer has to bound. For example, different intermediate acid transfer factor where it has to bound, that can be regulated by this trans-activity domain. Now your protein-protein interaction domain. So here the protein-protein interaction domain, which interact with the other transcription factor proteins, also proteins. So this is the three basic domains. Once again, I am repeating all those things: DNA binding domain, trans-activating domain, and your protein-protein interaction domain. Next is your this is some normal experimental approach with different culture media, with different bacteriophage mediated transcription factor being transformed, which gives rise to different kind of cells from a single type of cell. Ectoderm, mesoderm, endoderm, from the single mesodermal derivative cells, fibrous cells. This is what we call induced pluripotent stem cells. Transcription factor plays an important role to regulate those kind of cell differentiation mechanism. Here also some that kind of thing has done. So pathways. Okay. This is a mechanism of differential gene transcription. I will show in the next video. Thank you very much.